So I really want to express my very deep gratitude to the people for whom this award is named, the Liebers, and to the BBRF. And as Dr. Hirschfeld noted, NARCED has played a key, pivotal role in my career. And I don't want to belabor that. I'll talk more about that at the dinner tonight, about how NARCED influenced me. I'm going to be presenting research to you, research findings, examples of studies that we've done looking at the role of prenatal in utero risk factors for schizophrenia and for other psychiatric disorders. And this represents the culmination of over 2 decades of research from my lab. And again, it would not have been made possible without NARCED support, BBRF and NARCED support. So the question is, why is the prenatal environment important in psychiatric disorders? And that's really at the root of our studies. So it's known that autism, schizophrenia, and other psychiatric outcomes have a neurodevelopmental component. And the evidence for this is that before disease onset, we find that individuals destined to get these outcomes have delays in developmental milestones, including in speech, walking, and early social functions. And also, they have brain and structural and functional abnormalities. Second, prenatal factors are known to be related to adverse developmental brain outcomes. And these include such risk factors as infections, micronutrient deficiencies, smoking and environmental chemicals, and hormonal abnormalities. So we hypothesized that prenatal factors acting during fetal development are risk factors for psychiatric disorders. So we used a method called the birth cohort studies to address this question. This is an epidemiologic research design. All the work I'm showing you is from epidemiologic studies of large populations. And a birth cohort is a group of individuals who are born within a defined time period in a defined place and followed up for outcomes of interest. The birth cohort study is the most methodologically rigorous and informative approach to investigate prenatal determinants of later health outcomes. When we started our work, there had been previous studies that looked at pregnancy and birth complications in schizophrenia, but they had 2 key limitations. The first was the cases and controls were often assessed using retrospective interviews, asking cases and their mothers about the pregnancy history, rather than looking at the measures using what's second here, are biologically validated measures of prenatal insults. So our goal was to confirm measures of maternal exposures with biomarkers. These are biological measures of exposure. So I'll be using that word biomarker. That could be a biological measure. It could be from blood. It could be from a tissue, indicative of a biological process. And we wanted to confirm measures of these exposures with biomarkers during fetal life of the offspring who were later diagnosed with schizophrenia. And that was how we started our work. So I'll be describing a few studies where we looked at prenatal infection and schizophrenia. And this is how I started. Actually, my first NARCED was on this. So the first cohort I worked on was a birth cohort, all of whom were exposed to rubella in utero following the 1964 rubella epidemic. And I discovered this book by accident in the library one day called Psychiatric Consequences of Children with Congenital Rubella, which was conducted by Stella Chess, actually, who was at NYU at the time. And in it, she describes outcomes of individuals, all of whom had clearly diagnosed rubella in pregnancy, also known as German measles. So we found this rather remarkably high rate of schizophrenia and spectrum disorders. 21% of the cohort members grown up, we did psychiatric interviews on them, and we found that 21% had schizophrenia or a related disorder, spectrum disorder. And that was the breakdown by the different diagnoses. And that contrasts with the 1%, maybe 2% rate of the broader spectrum disorders in the population. So this then led me to get even more interested in infection, and I used another birth cohort called the Child Health and Development Study, much larger cohort, and one where we could look at a variety of risk factors, not only infection. So this included all mothers and offspring enrolled in the Kaiser Permanente Health Medical Care Plan in Oakland, California. There were greater than 19,000 live births, and the period of births was from the late 50s to mid-60s. So when we studied them, most had been through the age of risk for schizophrenia. And it featured a comprehensive database on a variety of prenatal and perinatal exposures. But most interestingly, there were archived maternal serum specimens, that is, blood specimens that were drawn during pregnancy. The serum were taken off, that's the non-cell component, where a lot of the interesting biomarkers are. And they were stored, frozen, and they're still stored for all these years. And what we did, and no one had studied psychiatric outcomes directly in this cohort, what we did is we identified individuals who had registry diagnoses and then confirmed the diagnoses with direct interviews of schizophrenia and bipolar disorder. And we identified a matched control sample. So this is the results funded by, and I'll be showing you examples of how NARCED, this is the NARCED Young Investigator Award. In this study, we looked at maternal exposure in pregnancy to influenza and schizophrenia in the offspring. And this shows the results for the first half of pregnancy and the second half of pregnancy. And for the first half of pregnancy, the prevalence of exposure to influenza in the mothers of cases was greater than the prevalence of exposure in the mothers of controls. So we found here a threefold increase in risk of schizophrenia if there was exposure to maternal influenza during pregnancy. And this was done by measuring influenza antibodies in these maternal serum specimens. In contrast, we didn't see a difference between cases and controls for the second half of pregnancy. So we next looked at toxoplasmosis, which is a parasite. It's quite common in the population, affecting greater than 20% of adults. It doesn't affect someone who gets it, the adult or even a child who gets it. But if a mother gets toxoplasmosis while she's pregnant, it gives rise to a variety of abnormal birth outcomes. And its sources include contaminated water, raw undercooked meat, gardening, or cat litter boxes. So there are many places where one can get toxoplasmosis. And this is also funded by NARCED, Young Investigator Award. And here we found that the occurrence of elevated toxoplasma antibody was greater, of high antibody, was greater in the mothers of the schizophrenia cases compared to the mothers of controls, about a 2.5-fold increase in risk. So now we've linked 2 infections in pregnancy with schizophrenia in their offspring. This is a little bit complicated, but I want to try to simplify it. So with our data, we're able to calculate what's called the attributable fraction. And the attributable fraction is the proportion of cases in a population that could be removed if we entirely eliminated the exposure. Now, it's not entirely realistic to totally eliminate it, but we still calculate this result. So we found if we could eliminate all of influenza in pregnancy, first half, we could reduce the incidence of schizophrenia by as much as 14%. If we could reduce toxoplasmosis, eliminate it, 13%. And this we also found an association with this type of infection, 6%. And because these infections don't overlap, we can basically add these numbers up. So, and again, not realistic, but if we could eliminate all these infections from the population, and they're playing a causal role, which we don't quite know yet, it's theoretically possible we could eliminate up to 1/3 of cases of schizophrenia from the population. Now, as I said, those are those caveats, and it's important to acknowledge, but even if we can reduce a fraction of these exposures in the population, it could lead to an appreciable reduction of schizophrenia in the population. And this has important implications for preventative approaches. We next move to looking at micronutrients. So these are things like iron and folic acid and vitamin A. They're all present in all our blood, and we need it. And we started by looking at iron deficiency. This is one of the ones. This is an example of what we've done. So the reasons we looked at iron were several-fold. Iron is involved in dopamine production, which is implicated in schizophrenia. And iron is important in the formation of myelin, an insulating sheath around nerve fibers needed to transmit electrical impulses between nerve cells. So here's called the axon. This is the nerve fiber, and this is the myelin sheath that surrounds it. And it was shown in an animal study, when animals deprived of iron in pregnancy, the offspring had reductions in this myelin. In addition, brain myelin deficits have been associated with schizophrenia in postmortem studies. So it gave us a rationale to test this. So here, they had already collected data on hemoglobin levels. Hemoglobin is a strong proxy of iron, and hemoglobin is used to diagnose anemia, so iron deficiency anemia. So here are the mothers with normal hemoglobin. Here are the mothers with kind of borderline anemia, and here are the anemic mothers. And this is the incidence of schizophrenia. The bars show the incidence of schizophrenia in their offspring. So we found that for the borderline anemic mothers, there was almost a twofold increased risk of schizophrenia. And for the anemic mothers, a nearly fourfold increased risk of schizophrenia. And it's adjusting for potential confounders of the association. So it's almost like a little dose-response effect here, suggesting the maternal iron deficiency may be important in the risk of schizophrenia in the offspring. But there were limitations of the CHDS birth cohort. First, we had a low occurrence of certain disorders that we wanted to study, like other disorders to study, like autism and ADHD. It still had a modest number of cases because schizophrenia is not very common in the population. I mean, there's certainly too much schizophrenia, but it's not all that common. So you need an extremely big cohort to get really good ability to show differences between cases and controls. And loss to follow-up, I won't go in great detail about, but there was loss to follow-up. So I started the study in Finland called the Finnish Prenatal Studies, or FIPS. Funded in part by an RSED Independent Investigator Award. And this is based on, I found this cohort, basically, called the Finnish Maternity Cohort. And in Finland, they've been collecting serum on virtually all pregnant women since 1983 in the entire country. So these maternal serum specimens are drawn in pregnancy, stored in a single freezer, and there are 1.5 million pregnancies. And these serum were drawn during early pregnancy, months 2 to 4. An important developmental period for the fetus. We then linked these, and what really made this study possible was not only this, but this. So in Finland, they have a socialized healthcare system, and they have computerized national registries, which are basically big electronic databases on the whole population in terms of all the diagnoses that they receive, including psychiatric outcomes, medical and birth registry on perinatal complications, and a population registry that can be used to link family members with each other. And we conducted this work largely in this city called Turku, Finland, and the biorepository is up here in Oulu, which is not shown on the map. So basically what this means then is that the country is the cohort. I mentioned the CHDS, that was a cohort in Northern California, relatively isolated cohort. This is the whole country, so it gives us nearly complete identification of all diagnoses of psychiatric disorders, including schizophrenia, autism, bipolar disorder, ADHD, and so on. Maternal serum specimens in this national birth cohort allow for measuring these biomarkers of the prenatal exposures in pregnancy long before illness onset. And we have availability of serum and diagnostic data on all subjects. And that allows us to apply the same methods in the same cohort to evaluate prenatal risk factors for different diagnoses. The large sample sizes provide a higher probability of detecting associations between prenatal factors, risk factors, and these outcomes, far larger than the CHDS cohort. In the Finnish prenatal study of schizophrenia, we have 1,500 cases. In the Finnish prenatal study of autism, we have 4,000 cases of autism spectrum disorders. Finnish prenatal study of bipolar disorder, 1,900 cases. And the Finnish study of ADHD, 10,000 cases. So we started by looking at maternal smoking and schizophrenia. Smoking during pregnancy is a significant public health problem. 12 to 25% of all pregnant women smoke. And nicotine readily crosses the placenta and the blood-brain barrier. Prenatal nicotine exposure is associated with disturbances in fetal brain development, and we measured maternal cotinine, a breakdown product of nicotine. We found here that the mothers of heavy smoke, the mothers who smoked heavily during pregnancy, the proportion of these mothers was greater if their offspring had schizophrenia compared to if their offspring were controls. There was a 37% increase in risk of schizophrenia following heavy maternal smoking in pregnancy. And that was identified by having high levels of this cotinine, this breakdown product. We also investigated maternal thyroid hormone during brain development and schizophrenia. The reason was, there were two reasons. One is that maternal thyroid hormone is correlated with fetal thyroid hormone. And secondly, thyroid hormone, this is the thyroid hormone is known to affect the development of brain regions and biological processes implicated in schizophrenia. And here, we looked at this condition called hypothyroxanemia, which is a condition characterized by low levels of thyroid hormone. We found an increased percentage of mothers with low thyroid hormone levels during pregnancy in offspring with schizophrenia in over 900 case-controlled pairs in this cohort. So here, there was an increase in the proportion of mothers of cases who had this hypothyroxanemia compared to the mothers of controls, or a 75% increase in risk. So we then moved on to look at autism. Now, why autism? Well, one, autism has a higher occurrence in the population than we had always previously thought. When I started getting funded, rates of autism were really increasing. And the question then was, if prenatal factors are related to schizophrenia, might they also be related to autism? So we looked in the FIPS-A at maternal factors in pregnancy and the risk of autism in the offspring. And we started by looking at C-reactive protein, or CRP. It's an established marker of inflammation and of activation of the immune system. It's substantially elevated following many different types of infection, and in animal studies, including those conducted by Dr. Kellendonk, who you'll hear from, activation of the mother's immune system causes brain abnormalities in the offspring. This may provide a reason that different infections during pregnancy are related to psychiatric outcomes in the offspring. We found increased maternal C-reactive protein levels during pregnancy, not only in autism, not only in schizophrenia, in both autism and in schizophrenia. So what these numbers mean, this odds ratio, 1.80, means that the odds of autism is increased by 80% in offspring of mothers with high levels of this inflammatory biomarker in pregnancy, and the odds of schizophrenia increased by 58% in the offspring. So this suggests that inflammation in pregnancy may be a common risk factor for both autism and for schizophrenia. And that isn't true for all psychiatric outcomes, but I wanted time to show you those data. We next moved to look at bipolar disorder with psychotic features, and I'm taking you back now to that CHD-Esper cohort. I had shown you earlier that maternal exposure to influenza was associated with the risk of schizophrenia in the offspring. Here, we looked to see if maternal influenza was related to bipolar disorder with psychotic features. And we first looked at diagnoses of clinical influenza, that is, influenza diagnosed by a physician. So these are the mothers of the cases who were diagnosed with the percentage diagnosed with clinical influenza, and the percentage of the mothers of controls who were diagnosed with influenza during pregnancy. We can see here a much higher rate diagnosed. And the odds ratio, meaning this slide mainly means that maternal exposure to clinically diagnosed influenza was related to a greater than 5-fold increased risk, greater than 500% increased risk of bipolar disorder with psychotic features in the offspring. Sorry if I said schizophrenia earlier, I don't mean to confuse you. We then measured antibodies, just like we did in the schizophrenia study. But these are the mothers of bipolar disorder with psychotic feature cases. And we found a nearly identical finding, confirming this clinical influenza finding with a greater than 5-fold increase in risk of bipolar disorder with psychotic features if there was an antibody-diagnosed influenza. These are other findings, and I won't have time to, in the interest of time, I won't present each one, but I wanted to show you that this was funded by an NARCET Independent Investigator Award. Low maternal folic acid and low vitamin A levels were shown in our study, and these are all from my group. There are many other groups that do this, but do this kind of work, although most of the work I've been showing you have been seminal findings. Low maternal folic acid, low vitamin A levels are related to schizophrenia. We found that maternal exposure to elevated levels of the insecticide DDT was related to risk of autism in the offspring. And this is really hot off the, not in the presses yet, but in preparation, funded by my NARCET Distinguished Investigator Award. We found, as I mentioned earlier, that low thyroid hormone was related to schizophrenia. Here we showed that low thyroid hormone, hypothyroxanemia, is also, in pregnancy, is also related to bipolar disorder. So now where are we going? So I know Dr. McGrath will be talking in more detail about prevention. I don't want to redo a lot of what he's saying, but I did want to make the point here, though, that this has the potential to lead to prevention of schizophrenia, as I alluded to earlier. So, for example, folic acid during pregnancy has been proven to lead to a marked decrease in neural tube defects. And many risk factors associated with schizophrenia, including certain infections, smoking, and anemia, are preventable with straightforward public health measures. This opens a window to the identification of new exposures. We're currently doing new studies in the FIPS, and we've done new registry linkages following people up longer and have doubled our sample size of cases of schizophrenia as well as the other outcomes. We can continue to look at diagnostic specificity, having all these diagnoses, which are done in the same cohort using the same methods. And we can look at gene-environment interplay. Now, so much we don't have genetic data, but this suggests that investigators in the field with genetic data and the environmental data can identify whether some offspring might be more susceptible to psychiatric outcomes if they have genetic mutations. In addition, understanding environmental factors may help identify new risk genes. And finally, animal models of prenatal factors, I think, are necessary to really get at the causal biological mechanisms. And that's work I alluded to that I've done with Christoph Kellendonk, Sarah Canetta, in a collaboration. This is called translational research. And one example of that is maternal infection or immune activation. One can then model other prenatal factors using similar methods. So finally, I'd like to thank my many collaborators in this effort in Finland, at Columbia, and at other places. And I really appreciate your attention, and I'm happy to take any questions.
You mentioned exposure to influenza as a higher risk for these other conditions. I'm just wondering, is that the live virus, or if you get inoculated during that time, does it increase the risk as well? And is there any information with if getting it in advance, or is there any effect in that regard?
Right. So the people who had the clinical influenza would have had the live virus. The measures that we did were antibody levels, so that's the IgG antibodies. And it's a long story, but what we did is we did a validation study that was funded by my first NARCET, and we showed that using a single antibody titer to influenza, we had samples in that pilot study where we had where we could show the gold standard for influenza infection diagnosis is an increase in the antibody titer by 4-fold rise. Now, we didn't have serial samples in most of the cases, so we used only one sample. But we had shown in that study where we had, these weren't controls, they weren't in cases, because we, as I said, we didn't have them in all the cases and controls. But we took people who had serum drawn from every trimester, so we could document 4-fold rise in titer, and then we looked to see whether there was a certain antibody titer that was predictive of whether or not there was a 4-fold increase in the titer. And we had about 80% positive predictive value, indicating a pretty strong relationship between the titer measures that we did and whether there was seroconversion to that influenza during pregnancy. We didn't use any data on vaccination, inoculations, anything like that. We didn't have data on that. We were interested in it. Does that answer your question? It's a long, sort of, okay. We don't have the data on the live virus. We couldn't measure live virus in the sera. Yeah, it's very hard to do that.
Ellen, you mentioned that the finding of with the attributable risk for bipolar disorder with psychotic features was like 3.5 times it, which is a lot higher than for schizophrenia and autism, as I understand it. That's a really striking finding, if I understand that correctly. Is that?
It was actually a 5-fold increase in risk for influenza exposure, and it really is stronger than what we found for schizophrenia. It's a little tricky to make these comparisons because, again, I didn't have time to show it, but
Sure.
We have what they call confidence intervals. So the confidence interval is like how confident are you that a level is between one and another. So it's hard to say that it really is definitively higher, but at least these data suggest that the odds ratio is higher.
That's amazing. Yep. A couple here.
Specific data for postpartum illness that, it's a pretty specific illness. And then the products of the mothers who had it.
The products of the mothers?
The products of the mother. No, the child who grows up who was a product of postpartum.
Oh, postpartum. You mean postpartum depression or
Yeah,
psychosis
yeah,
or
yeah.
other? Right. So we can look at. We have not looked at that yet. We can look at that in Finland because we do have diagnoses of postpartum psychosis and depression, and I have an interest in studying that. It's one of the things that we just haven't had time to get around to yet. But I think that could be quite important, and it's interesting in the sense that there are different explanations for why that might be important, and that how it might affect especially the development of the child. Might the child be more likely to get depression if her mother had postpartum depression or other outcomes? So that is something that we hope to do.
Yeah, really.
I've heard about like the myelin deficiency, the breakdown. Doing studies on that. Do you think they will ever, like MS drugs, like natalizumab? Do you think they will do more trials with that, or is?
Do I think they'll do more
Like trials.
trials of
for
tocilizumab?
Yeah, for the myelin breakdown, the myelin sheath breakdown. Do you think that will ever go far, or no?
So an idea might be to give tocilizumab to prevent the breakdown of myelin? I mean, it's an idea. I mean, the issue is giving that any drug in pregnancy, one needs to be careful that the drug doesn't have a teratogenic effect. So the offspring have already had their myelin broken down when they're born. The issue is, can somehow that be reversed later on? Is a question. I'm really glad you asked that question because I did another study with a guy named Raghi Girgis at Columbia, where we gave it was his brainchild, and we looked at tocilizumab and given to chronic schizophrenia cases, and we found no effect for the tocilizumab. The issue there was that we were testing an immune hypothesis. So the idea was it reduced interleukin-6, which is another inflammatory biomarker in the blood. But I like your question. I like the idea too that there might be certain other interventions that could affect those downstream biological processes that are induced by these prenatal exposures. So thank you for your question. Thank you to everyone for your questions, by the way. But
Way back there, yep.
Herb, do you have a question, Herb? Oh, someone else?
I have a question about the smoking during pregnancy. Does being exposed to secondhand smoke have any effect?
Yeah, so we don't, that's a really good question, really important question, but we don't, so the cotinine levels in the mother could have come from secondhand smoke, and it would be interesting mothers did answer questions about if they smoke or not, but they're often not always honest, for a variety of reasons. So the issue was if we really had good data on that, if they said they didn't smoke but had increased cotinine levels, then we could find out if the partner smoked and look at whether it was secondhand. So these cotinine levels don't tell us if it's firsthand or secondhand, but it could argue that if you do get cotinine, nicotine in your blood from secondhand smoking. I don't know enough this literature, and I defer to other people, but I would think that levels of cotinine or nicotine in the blood from secondhand smoke would be lower than if you smoke yourself. And our finding, as I mentioned, was specific to the heavy smoking group. So I'm suspecting that it was more of a the mother smoking herself. But I think that should be further investigated by doing interviews and assessments of secondhand smoke. That's a big problem.
Dr. Partis?
To what extent has the awareness or the evidence showing that there may be environmental factors involved in the frequency of schizophrenia had on actual practice and the care of people in general? How seriously has it been taken, and what things do people do in order to address that?
Right, so I think that's a question. I don't think enough is being done on that. I will say first that a number of these risk factors are being prevented anyway, because now more moms are getting influenza vaccines, better attention is given to giving dietary supplements, but I still think it's a significant problem. What our hope is that this can get communicated further to people in public health, to obstetricians, gynecologists, and of course to the mothers themselves while they're pregnant, to see if it's possible to further prevent the risk by giving a more enriched environment to the fetus in terms of nutrients, by avoiding certain infections, and there are very simple, straightforward measures that can be done to prevent that, and certainly non-smoking, avoiding smoking, and that's a significant problem. So I think a lot more needs to be done in terms of encouraging these groups to avoid these various behaviors. And it's not to say it's the mother's fault or anything like that, because the parents, they don't know that what these are doing to the offspring. So that's not the idea. The idea is to reduce the incidence of these serious psychiatric conditions.
Ellen, thank you.
Thank you.